I'd like to thank everyone for giving me the opportunity to present today. For the past 15 years, I have been passionate about understanding how our cardiovascular system develops and what goes wrong in the context of cardiovascular disease. Many of you may know that heart disease is the #1 killer of adults in America. But what you may not know is that heart defects is the #1 form of birth defects in children born in America. This is an incredibly important condition because nearly 1 in 100 children are born with some form of heart defect. And despite all of the latest technological innovations and all of the advances we've made in genomics and AI, for over half of the cases of children born with heart defects, we actually don't have an attributable cause. So my lab has taken an approach where we take stem cells from children with heart defects and combine that with mouse models of congenital heart defects. We use the latest technologies with CRISPR and artificial intelligence, looking at not just one gene, but at every gene in our genome. And we ask a basic question. What are the different cells that are responsible for the development of the heart? And what are the cell types and the mechanisms of the defects that lead to congenital heart disease? I want to give you one story of what my lab has been working on recently. We are interested in understanding why children born with Down syndrome are born with heart defects. Down syndrome is an extraordinarily complex disorder that affects multiple cell types and multiple organ systems in development, including neurological development in the brain, craniofacial development in the head, neck, and face structures, as well as the musculoskeletal system. In particular for us, nearly half of children born with Down syndrome are born with heart defects. The specific kind of heart defect they're born with most commonly is called a septal defect. A septal defect happens when the walls that normally separate the 4 chambers of our heart have a hole that allows blood to flow from one chamber to the other. Children with Down syndrome are at high risk for being born with a complete atrioventricular canal septal defect, meaning that the walls that separate all 4 of their chambers basically isn't even present. Now, we know what causes Down syndrome. Down syndrome is caused by having 3 copies of chromosome 21 instead of the normal 2 copies, okay? But what we don't know is what is it about that 3rd copy of chromosome 21 that disrupts heart development? We started by asking a specific question. Are there genes on chromosome 21, not the whole chromosome, but specific genes, that when you have them in 3 copies are bad for the developing heart? What we did was we first asked, what are all the genes on chromosome 21? There's about 230 genes on chromosome 21. We then used stem cells from a child who had Down syndrome, and we integrated both CRISPR editing technology and the tools of machine learning to ask the question, if we could boost the levels of each one of those chromosome 21 genes to 3 copies instead of 2 in a normal cell, can we convert a normal cell to have the transcriptional and gene expression profile of a Down syndrome cell? And when we did that, we zoomed in on one gene on chromosome 21 called HMGN1 that we thought was responsible for heart defects. And so what we did was we took then a mouse model of Down syndrome and we performed micro-CT analysis where we could look at the 3-dimensional structure of the developing heart in this mouse model of Down syndrome. Here I'm showing you a healthy mouse heart where you can see the walls separating the 2 ventricular chambers. Here I'm showing you an image of a Down syndrome mouse heart where you see that there's now a hole separating the left and the right ventricle. And when we reduce the copy of the gene that we identified, HMGN1, back to 2 copies in this mouse model, we were able to seal that gap and restore the incidence of septal defects back to healthy levels again. So my lab's fundamental mission is to try and find new treatments for pediatric disorders and diseases. Our goal is to understand the cause of congenital heart defects, to try and reduce the size of that pie in which we don't know what is happening in these children with congenital heart defects. The innovations that we're using are that we're merging both experimental approaches and all of the latest computational tools. And our goal is to develop new treatments for heart defects for children, and we're particularly excited to do this in collaboration with clinician scientists and friends at Rady Children's Hospital just down the street in La Jolla.